So I'm going to pass this to Ken, who has, CD2H is really a partnership between the government and academia. And Ken has been a genuine leader in this partnership and will highlight some of the hashing data. Ken.
Thank you, Chris. Can you all hear me okay?
Yes.
So I don't know if everybody is familiar with the technology called Privacy Preserving Record Linkage, or PPRL, sometimes known as hashing. Because of the terrible crisis, excuse me, we've been able to reach high levels of the NIH about the possibility of funding this technology across the entire NIH. And the idea is that this hashing or technology would allow us to do lots of things that are really important and facilitate research. One of the most basic things is that it will allow deduplication of patients. We're not going to go into the details today because I really want to show this demo, but it's very important for data quality. The other thing is what we're going to show in a few minutes about linkage. So think of different types of data where you would want to integrate an image data or with the N3C data or waveform data or anything. The other thing we can use this for is cohort discovery, very much similar to what the RICs are doing with Trinetics and ACT. And so this would just be another tool in the quiver, so to speak. And then finally, the ability to link observational data. So next slide, please. So what we thought we'd show you a short demo today. We would love to show you more when we have more time. But we're going to have Joel Saltz on from Stony Brook, Fred Pryor from TCIA, University of Arkansas, Pratik Prasanna also from Stony Brook to talk about imaging, and then Andrew Gervin, Chief Scientist at Palantir, to actually show us the demo and the work they've done. Next slide, please. So again, what we're going to show you is that we have linked imaging data of the same patients that are also in N3C. So the idea is to be able to, look at images or query images or N3C and then to see a visual correlation with the radiographs. So with that, I'm gonna, if we can stop sharing my screen, and we'll hand it over to Andrew. Or I guess it's Joel, if you want to introduce something.
It's great to have an opportunity to talk to this group, and the promise of hashing or linkage between disparate data sets maintained by different institutions or different consortia has been clear for years, and it's been, I think, frustrating to many of us who work actually in multiple groups looking at sort of different views of data, often coming from the same patients. And certainly image analysis, radiology, and for that matter, pathology analytics often is quite data poor when it comes to understanding clinical trajectories. And most sources of electronic healthcare data don't really contain imaging, at least at a consortial level. So this has certainly also been the case in COVID, and the multi-organ system impact of COVID, as well as the need to predict outcome and who needs to be treated more intensively. Both are strong reasons to link imaging. So our group has taken this on, N3C and the imaging group, as one of our pilot hashing demonstrations, as well as will soon all being well become a mature project. So Fred Pryor, who is the lead for the Cancer Imaging Archive, as Ken has said, is aggregating lots of de-identified COVID images from our place, is one of the places, Arkansas, and a number of other folks. We're also working with RSNA and MIDRC, which is a large consortium dedicated to COVID imaging, and linking the image data sets from those places. Initially, what you're going to see is just Arkansas, with the N3C data using the mechanisms that we'll be talking about. So that's what the demo is. Pratik Prasanna is at Stony Brook, as Ken mentioned, and has been doing a lot of great work on radiomics to use imaging to predict COVID course, as is actually Fred Pryor. So Pratik is going to talk about some of that work as one of the several motivating examples. So, but next is the demo from Andrew, who is Chief Technology Officer. So hit it, Andrew, for Palantir.
Okay, great. Can everyone see this?
Yes, we can.
Okay, perfect.
And Andrew, before you start, I do need to say that this is all publicly available information right now, so we're not sharing any private information. Sorry, just wanted to put that disclaimer in that.
Yes.
Thank you.
Absolutely.
Sorry, Andrew.
So this data has been linked, but we're not going to show any of the N3C data for these patients. All right, so I'm going to take you on a quick demo of the integration of the imaging and the clinical data inside the instance of Palantir Foundry that N3C is using. The Foundry platform has already been used in many different biomedical R&D applications with clinical, genomic, imaging, HTS, and other data types. So in this particular instance, we have close to 600,000 COVID positive patients from 39, and as of tomorrow, 40 sites that have passed data quality control. And for these patients, we have about 2 years of EHR data. And now for a small subset of them, we have linked images from the TCIA. And of course, we hope to expand this greatly using the hashing technique that Ken and Joel discussed. So in this interface on the TCIA public data, we have information about the cohort of those patients that are in both systems. We have their demographics, so sex and race, their height, weight, and BMI, and then we have outcomes such as ICU admission and mortality. All of these patients, as we mentioned, are coming from Arkansas for now, but soon we'll have Stony Brook and other sites. So if we actually take a quick look at the patients who were not admitted to the ICU, we can take a deeper look at one of these lung radiographs and see that it is relatively clear. And if we go down and look at the radiologist findings, we see a lot of examples for the different patients. The lungs are clear, the lungs are clear without focal consolidation. Otherwise, the notes are very positive on this group. If we contrast that with the set that were admitted to the ICU, we can see a pretty different profile. I'll just pick one of these, where the lungs are much less clear. And if we look at the radiologist notes, we see subtle streaky opacities, hazy opacities, low lung volume, etc. Down below, we have information I'm not going to show today about the labs and the time course over the hospitalization for these patients, but we won't show that because it is not public information yet. So the obvious question that you might have, given that we are already building models based on labs on admission that can, to a good degree of accuracy, predict the severity of a patient's COVID disease, we might want to add the imaging data to really reconstruct that patient, since the lab data doesn't give us the full picture of what's going on, and that should hopefully improve the quality of these machine learning models. So to get at that, we used some routines from Pratik Prasanna at Stony Brook to first segment the images, to isolate the lung tissue, and then to calculate a series of radiometric quantitative features about each of these samples, and then compare across those patients that were admitted to the ICU and those that weren't. There are many features here. Here's an example of just one of these features, the original first order skewness. It is significantly different between those that were not admitted in orange and those that are admitted in blue. If you show two features here, you can begin to see that you're separating the population based on these radiometric features between those that were admitted and those that were not admitted. And so the idea would be to expand to all of these features and build a complete and accurate classifier. And then combine that, of course, with the lab data. And to do that, we need many more images, and that's where the hashing will come in and hopefully allow us to get up to the thousands or tens of thousands of images. So questions about that? Otherwise, I'll stop and hand it off.
Okay, so Pratik, I think is now you're up.
Yeah, so Ken, could we have the slides back on, please? Great, thanks. So as we heard from both Joel and Andrew, imaging has proven to be a very important tool for disease quantification and staging, and this is extremely vital for anticipation, such as availability of ICU beds, patient management planning, as well as to accelerate, let's say, drug development through reproducible and quantified assessment of treatment response. So as a step in this very direction, we at Stony Brook University have been working on developing and validating machine learning tools to predict ventilation requirement and mortality from baseline chest radiographs and non-imaging clinical parameters like oxygen saturation data. In one such model that you see in this slide, we first train a deep learning algorithm to identify the different lung fields. So we train this model to leave out the the different artifacts such as EKG leads and so on. So this is followed by subvisual characterization of the lung regions using radiomics. These features, like Andrew mentioned, may not be visually discernible, but can be derived using different computer vision techniques. Finally, we use these features in conjunction with machine learning and deep learning classifiers to predict different outcomes of interest. The box plots that you see on the slide, they depict radiomic differences between different outcome groups. Next slide, please. So here we show some results that we have obtained using a pilot multi-institutional data set. In the top left panel, we can see lung segmentation results using our deep learning models. These models have been trained on de-identified scans from Stony Brook University and have now been validated on TCIA. So these ensemble techniques were also able to automatically identify regions of prognostic importance on the X-ray scans that you see on the right side of the screen. The sensitivity and specificity of prediction for ventilation requirement using just baseline chest radiographs was computed to be 90% and 71% respectively. The scatter plot that you see on the bottom right half of the screen, it shows a visualization of the deep features ability to separate different outcome groups. What we have seen here are chest radiographs, but integrating but imaging is also getting increasingly important, where we are integrating other modalities like CT, for example, where infiltrates such as ground glass opacities and consolidations are more discernible. So keeping in line with our theme of longitudinal multimodal data integration, the N3C TCIA linkage that we have heard from Joel and from Andrew, it provides us a unique opportunity to develop these automated machine learning based quantification schemes using ensemble of architectures and a data-driven consensus for diagnosis and outcome prediction of these patients by fusing imaging biomarkers with other non-imaging clinical and biological attributes. Thank you.